So when we think about growth hormone that we prescribe regularly, it's a 22 kilodalton protein with 4 alpha helices and 2 disulfide bonds that binds to the receptor dimers. So one growth hormone molecule to 2 dimers of the growth hormone receptor. And so as growth hormone is modified, we have to make sure that it is still able to interact with the receptor to have its activity. Currently, growth hormone lasts, when we think about the native molecule, it lasts 14 to 19 minutes after an IV injection. So on this slide, you can see the rapid clearance of growth hormone after IV injection, and a much more prolonged half-life of 2.1 hours after subcutaneous injection. But when this compares to the 24-hour serum profile, if you look at growth hormone as a daily injection in this area under the curve versus the pulses that we typically see for growth hormone, it's definitely daily growth hormone is non-physiologic. So why would we think about having a long-acting growth hormone? First of all, in the previous slide, I talked about the physiology. So is there a potential to make a growth hormone more physiologic? And most of what I will talk about today will not be more physiologic, actually maybe even less so. But I think the main reasons are going to be the potential for improvement in adherence, persistence, and the growth response. So by making a medication less frequent, can we make it more likely that people will take it and have a better response to that medication over time? And I think the key question, or the key point that's made here by both Hippocrates in 400 BC and C. Everett Koop in the last century, was that drugs don't work if you don't actually take them. And so we need to make sure that people are taking their medication so that it helps. And the administration of burden of daily growth hormone therapy has been well established. I'll talk about a few studies today that look at both patients and caregivers' perception of the burden of growth hormone therapy. And in the patients, the storage requirements were a significant issue because when you travel, you were likely to miss a dose because you couldn't have it outside the refrigerator. And parents had similar concerns about the medication, but also the actual injection, bruising, painful, causes stinging. And so, patients who have to take an injection every day are going to experience these effects more frequently. When we look at the best compilation of data of non-adherence, it's 3 different studies, one from the United States using the Growth Hormone Monitor Registry with a EasyPod device that is electronically capturing compliance, a New Zealand study by Cutfield that's looking at actual return of the vials and counting the vials to describe how people are using it, and then observational longitudinal study in Spain that all show that the more growth hormone injections that you miss, the less well you grow. And they also document that there's a significant amount of medication missed on a regular basis in our patients, more so than I think we realize. We looked at the Novo Nordisk Answer program to look at also how long do people stay on the medication and why do they stop it? And the reasons for discontinuation in that registry included final adult height. Final adult height was achieved at about 35% of the time. Patients and the caregivers agreed about 16% of the time that they were done, but about 4% discontinued because of lack of adherence. About 1.5% were lost to follow-up, which is also likely lack of adherence. And in the US, a number of insurance issues were also part of why people didn't take the medication. But not only taking it every day, but staying on the medication is going to have the long-term impact. And then when we reviewed, looked at 5 relevant adult growth hormone adherence and persistence trials, there was actually pretty good adherence rate between 70% and 91%, but there was a high treatment discontinuation rate, depending upon the study, anywhere from 13 to 58%. And in adult growth hormone deficiency, the lack of awareness of the growth hormone health benefits, the lack of feeling any different in terms of their own health status and quality of life were significant reasons why people stopped, but also the low adherence was a significant issue. And so low persistence with drug holidays was also key. So we feel that with daily growth hormone, adherence and persistence are a significant barrier to treatment. And the goal is having a longer interval between injections could improve adherence and ultimately the response to treatment. The other thing that, as a pediatric endocrinologist looking at height outcomes, strikes me is that we haven't quite hit the target of achieving a near adult height at the mean of the general population. And possibly that's because this isn't all corrected for initial height versus final height, isn't all corrected for their genetic potential. And we know that many parents of children who are short are also short. But I think that we're still missing some efficacy in the current daily treatment regimen. So what are the characteristics of an effective long-acting growth hormone? We want it to provide all the benefits of a naturally secreted growth hormone. We want the free growth hormone molecule to get to all the target tissues. We want it to be easily administered with a small needle in an injection device that works well, room temperature stable, pain-free, and lack of immunogenicity. So those are the ideals of what would the long-acting growth hormone look like. And in 2016, there was a Growth Hormone Research Society perspective on long-acting growth hormone to describe, as a community, what did we think was necessary to show that long-acting growth hormone would be effective and safe in the populations that we're interested in treating. And so they recognized that the long-acting growth hormone could represent a significant innovation, but that it was essential to be carefully surveilled in long-term studies. So important advancement, but we still need to know how it's going to help kids. That it reduced adherence to daily injections currently likely compromises the response to treatment. And then they recommended that because the different molecules have different pharmacokinetic and pharmacodynamic characteristics, we're actually going to have to develop safety and efficacy and dosing profiles that are specific for each molecule. And I'll talk about that later as well. In kids, the 1-year duration of treatment for naive children was an appropriate trial as recommended by the group. And in adults, the 6-month trunk visceral fat reduction as the primary endpoint was key, with dose titration, which is typically used in adults as part of the trial design so that it could mimic what our current practice is with daily growth hormone. So another question that comes up is whether or not long-acting growth hormone will actually be superior to daily growth hormone. Each of the molecules is being studied in phase 3 trials as non-inferior to daily growth hormone because of the pathway, the approval pathways that are available. But it's if we actually improve adherence and improve efficacy with superior IGF-1 levels and potentially superior metabolic actions, then we could see actually better height and metabolic outcomes in the long term with these new molecules. And so I think that although the trials are targeted at non-inferiority, over time, my hope is that long-acting growth hormone will actually be superior. So one of the key questions is, how do we make an injection of growth hormone last longer? And I list a few different ways that we can do that. First is to delay the rate of absorption from the subcutaneous space by incorporating it into microspheres or other methods. We can reduce the rates of clearance from the circulation, and there are a number of different ways that we'll talk about, including pegylation, both permanent and reversible, by adding a chemical entity to the molecule to protect it from degradation. We can modify its ability to bind to serum proteins such as albumin so that it will stay in the circulation longer. And then by fusion proteins, either using synthetic polypeptides or naturally occurring proteins. And there's some examples there that I'll talk about specifically of how we can change the molecule to make it last longer. So one of the first attempts to make growth hormone last longer, subcutaneous injection of growth hormone last longer, was using pituitary growth hormone, and it was actually using intramuscular injections after it was reconstituted into 15% gelatin. And in this paper, they talked about the lack of physiologic profile of daily growth hormone. And this was an attempt by stretching out the absorption of the molecule from the subcutaneous tissue to actually try and mimic more of a physiologic daily profile. And so Dr. Lippi and her colleagues did a very small study with 3 children. And on the left, you can see with the aqueous growth hormone that we peaked early and then had very little growth hormone present after 12 hours. With the resuspension of the medication into gelatin prior to IM administration, the peak was smaller, but it lasted a little bit longer. So they felt like a slower increase in the blood levels and a lower but more prolonged peak would actually give a reasonable response. And interestingly, in this small study, they then did the same reconstitution, but gave it 0.1 units per kilogram 3 times weekly, which was the recommendation at the time, and saw in a small number of children that there was actually no difference in the rate of growth between the two methodologies. And so interesting, but not pursued over time. The previously marketed molecule was actually Neutropin Depot from Genentech for pediatric growth hormone deficiency. This used microspheres to make the medication longer acting. So after subcutaneous administration, growth hormone was actually released from the microspheres as a burst and then a slower process over time. And so diffusion from the microsphere surface, followed by hydrolyzing the copolymer and releasing it into circulation over time. This was studied in both phase 1 and 2 and then phase 3 in children with pediatric growth hormone deficiency as a once a month and twice a month molecule or administration. And then as it went into phase 3, it was twice a month and once per month. And I wanted to focus, these numbers will become relevant later, of 7.6 centimeters per year in the once monthly group and 7.9 centimeters per year in the twice monthly group, which was slightly lower than what we would have expected in a daily growth hormone population at the time. The single dose of Neutropin Depot actually showed a profile in terms of coming down to baseline levels after about 2 weeks of injection, which is why it made sense to do the twice monthly injection. And when we look at the IGF-1 levels, we can see a peak of the IGF-1 in the early phase that comes down to baseline that differs slightly in the two groups, with the 1.5 milligram once per month having actually a lower peak. It was also studied in adult growth hormone deficient patients, and this is showing the growth hormone profile in men and women. So there were differences in the response between men and women between the two doses of 0.25 mg per kg per body weight and 0.5 mg per kg per body weight. But again, we're seeing that after about 10 days, we see the growth hormone level back down to definite baseline. And even at 7 days, we can see that it's already coming down. This is the IGF-1 levels in the adults, showing some persistence of the IGF-1 levels in the higher dose group compared to the lower dose group, and a similar pattern in women, though lower responses in women in terms of the IGF-1 response. Based upon these clinical data for the pediatric group, Neutropin Depot was approved in 1999 with the microsphere technology. But interestingly, after only a few years of being available on the market, there was a decision between Genentech and Alkermes, who developed and marketed the medication, to discontinue it. They reported that it was based on significant resources required by both companies to continue manufacturing and commercializing the product as the reason. But I think it was also a very painful medication that had lower growth responses than daily, and that definitely could have contributed to its continuation in the market. There have been a number of other molecules that have been started development and ultimately discontinued development for various reasons that I just want to briefly go over. A depot formulation by Altus Pharmaceuticals that was protein crystallization technology. 3 pegylated molecules, one from Ambrex that had some concerns about vacuolization of the choroid plexus in animal models that rose concerns about potential side effects in children. In the Novo and the Pfizer studies, there was discussion of lipoatrophy in some of the patients who received the medication, but it's not clear that that was why the medications were discontinued. And then the TEV albumin product that was being studied, fusion with albumin, was discontinued. There were some potential concerns about anti-drug antibodies, which will come up as a potential complication. Another product that I was very involved as a clinical investigator in that I want to just briefly highlight was Somaveritan. And Somaveritan was a molecule of growth hormone modified by nonsense sequence of amino acids on either end of the molecule that increased the solubility and stability of the growth hormone molecule, and was being administered twice monthly, and was a very large molecule at 119 kilodaltons compared to the 22 kilodalton native growth hormone size. And Somaveritan went through phase 1 and 2 with promising results, and unfortunately, as the phase 3 velocity trial results became available, it was demonstrated that in children with pediatric growth hormone deficiency, that they did not meet the primary endpoint of growth. And so in those children who had received the 3.5 milligrams per kilogram twice monthly, the growth of children was 9.44 centimeters compared to 10.7 centimeters per year in those receiving the daily comparator of genotropin. And it didn't meet non-inferiority because of the variability of the growth in the children and the mean value. And so treatment with this medication was discontinued. And unfortunately, the results of this trial haven't been published so that we could understand whether there were anti-drug antibody or other potential side effects that could have been involved in this outcome. But it demonstrated that this particular molecule, given twice monthly, was not effective in treatment of children with growth hormone deficiency. It also raised the question posed by Paul Sanger at the International Pediatric Endocrine Meeting in 2017. Is it because the molecule was larger, 5 times larger than daily growth hormone? Growth hormone's size has actually been very well conserved through evolution, and the size of the molecule may not be able to penetrate the growth plate or other target tissues such as fat, liver, etc. And so are we actually seeing an IGF-1 effect, but not a growth hormone metabolic effect? And Dr. Sanger actually brought up the question of, in the trial, were they actually seeing increase in BMI in children who were on somaveretan different from those results that have been previously shown in daily growth hormone registries of basically staying stable BMI, or in the data that was available at the time for TransCon growth hormone, where there was no BMI change in the children, compared to a 1 kilogram per meter squared change, or a 0.64 SD gain in BMI in the children receiving somaveretan. And so it raises an important question of, does the size of the molecule matter in its ability to reach the target tissue? I'll segue now into the current products in development or currently marketed, and what's the data that we have available to us to say how is it doing and will it be beneficial for children. It includes one depot molecule that's currently approved in Korea and had been approved in Europe, but has never been marketed, called Utropin Plus. Another depot molecule in preclinical studies, CP016, Jintrolong, which has been marketed in China now for a number of years and actually has some long-term extension data available for it, once weekly. A once-weekly pegylated molecule from Boulder Biotech that's in preclinical studies. TransCon GH, which is a reversible pegylation of growth hormone that I'll talk about in detail with phase 3 data for children given once weekly. Somapacitan with non-covalent albumin binding once weekly. Somatrogon from OPCO and Pfizer with once weekly treatment. And then several that are still coming with GXH9 once weekly or once every 2 weeks, Alt-P1 once weekly, and Profuse GH, which are once monthly, that are in the early developmental phase. And I won't talk about the last two any further. This is just a cartoon to show where things are at in the process of coming to market. As I mentioned, Jintrolong already has a number of children on treatment commercially being followed in extension studies. They're looking at adult growth hormone deficiency as well as other indications of treatment. And I think the indications here is important because we want to see other indications ultimately be studied. And currently, somapacitan also has SGA as a clinical trial that's ongoing. And I think it's going to be important to see those indications evaluated. And I do want to point out that there's also a concern about the failed phase 3 trial in somatrogon in terms of its adult efficacy outcome, but promising data from the phase 3 trial in children. So as I go through each molecule, I'll go a little bit into the methodology of how it works and how it's released. So Utropin Plus is native growth hormone, 22 kilodaltons. That's a depot formulation in sodium hyaluronate microparticles. So it's slowly released into the tissue. The phase 3 trial for pediatric growth hormone deficiency showed 2-year treatment results. So baseline growth velocity, you can see here that 0.5 milligrams per kilogram per week versus 0.03 milligrams per kilogram per day of genotropin. And then during their extension, they transitioned to the 0.5 milligram dose. So 12 months on one or the other, and then 12 months just on the investigative product, we can see 11.6 versus 11.97 for a first year height velocity. And you can see the height standard deviation gains were similar in the two molecules, showing non-inferior response to treatment. Gintralong is a irreversibly pegylated molecule of approximately 62 kilodaltons. So basically attaching the PEG to the growth hormone at amino groups to try and prolong its activity. Phase 3 studies of Gintralong, they did baseline to 25 weeks in 0.25 mg per kg per week of daily growth hormone versus 0.2 of the Gintralong. Excellent 13.4 centimeter growth velocity response, but similar to the daily growth hormone treatment. Adverse events were actually comparable between the two groups, with headaches, injection site reactions, and hypothyroidism. So it was statistically superior to daily growth hormone, with both the height velocity and the height velocity increase, with a similar safety profile. And as I mentioned before, there are a number of phase 2 studies for Gintralong in different indications currently underway, as well as phase 4 studies that I learned about when I visited China earlier this year. Somapacitan is being evaluated by Novo Nordisk as a long-acting growth hormone molecule with a single point mutation that has a side chain terminal fatty acid with non-covalent albumin binding properties, which improve the binding to circulating albumin. And this cartoon just shows the growth hormone molecule binding to the growth hormone receptor with the fatty chain sticking out here. It's not drawn to scale. It's 23 kilodaltons, so this is a very small change to the molecule size, and has been, this is the, again, not to scale, the albumin linker that has already been shown in other molecules that have been marketed by Novo Nordisk to be successful in prolonging the activity of those molecules without changing the safety profile. And so I think this mechanism of growth hormone prolongation has promise for being a safe method that doesn't really change the either efficacy or safety of the medication, which of course we have to then prove in clinical trial. So far, somapacitan is in phase 3 studies in children with pediatric growth hormone deficiency currently. So we have phase 2 data that we can evaluate as to the safety and efficacy. And in red in this slide, we can see the 52-week annualized height velocities in the lowest group, in the lowest dose of somapacitan, 7.8 centimeters, 0.08 has 9.7, and 0.16 has 11.5. So we do see a dose response curve showing that higher doses are increasing the growth response. And ultimately, compared to daily growth hormone, it was superior in response at 52 weeks. We also have IGF-1 data showing the peak and trough values that we're getting to average at 26 weeks with the 0.08 dose and slightly above average for a peak value or +1 standard deviation versus daily we're getting to the mean. And a model-derived average is showing that it's just above, with the highest dose in the phase 2, that it's just above the mean, which would be an ideal goal. The other thing is that there are some peaks greater than +3 standard deviation. In adults, this was studied with placebo and somapacitan versus daily growth hormone, and then daily versus daily with the washout period. And this is a busy slide, but it shows the IGF-1 values. As a group, you can see that they come up into the normal range, which is the goal for therapy, quite nicely in the different groups. And then when they continue or transition to the other molecule, that we have IGF-1 values in the target range with the treatment as well as with the daily growth hormone. Somatrogon, I mentioned earlier, is developed by OPKO and Pfizer. It has 2 CTP molecules from the natural human chorionic gonadotropin on one end and one on the other end to prolong the circulatory half-life. These molecules are actually part of why HCG lasts longer than LH in the blood. And so naturally occurring prolongation, it does increase the molecule to 47.5 kilodaltons. This is the phase 2 data, which shows multiple different doses of somatrogon compared to daily growth hormone, seeing increasing height velocities in those groups in response to treatment. And I want to jump to the phase 3 data, which was presented last year in a press release, but also by Sherry Deal at a recent Endocrine Society presentation that showed it was statistically superior, numerically superior to growth hormone by 0.33 centimeters per year. Transcon GH by Ascendis is native growth hormone that is chemically connected to the transcon linker that is then hydrolyzed and releases native growth hormone that is unmodified into the body. In their height trial presented last year at Endo, comparing daily growth hormone with genotropin to the transcon GH, it was not only numerically but statistically superior at 10.7 versus 9.7 centimeters per year. In more recent data that was presented by Dr. Maniatis at Endo recently shows that younger children responded quite nicely to transcon GH as they were converted from daily to weekly transcon therapy. Interestingly, they also did see a number of elevated IGF-1s in both groups, both receiving daily growth hormone and those children transitioning to weekly therapy. And so I think that's an important thing that we need to monitor of how do we measure growth hormone effectiveness? Do we measure the IGF-1 level? And when do we measure that IGF-1 level to determine the best dosing regimen and the best outcomes for the kids? I'm only briefly going to touch on GXH9. It's a hybrid IgD-IgG fusion protein that's 100 kilodaltons, proposed every other week dosing schedule. We have data in pediatrics, the phase 2 that was presented about 2 years ago. It doesn't really show a dose response to the different doses of GXH9, but it was similar to the comparator medication. Even though long-acting growth hormone is in its infancy, there's already a meta-analysis published last year showing that the height velocity was similar between the studies that were analyzed. The height standard deviation changes were similar in the studies that had been so far analyzed. When we look at the IGF-1, because the reports actually give both peaks and troughs, they were slightly higher in long-acting growth hormone patients. So if we think that high IGF-1 is bad, it favors daily. And if we look at adverse events, the number of adverse events favors the daily growth hormone compared to the long-acting growth hormone in what has been reported thus far in clinical trials. And so I think we're still, of course, getting more information about growth hormone therapy and long-acting growth hormone. But at this point, we have some very promising data showing that kids are growing well with treatment and that there haven't been any new treatment emergent problems with the long-acting profiles so far. So the key points that I want to give today is that daily growth hormone requires daily administrations, which impact adherence with frequent dose omissions and treatment cessation. We have a number of different technologies that have been attempted to prolong growth hormone action, but none are approved yet. What we've seen so far has had some being discontinued to a lack of efficacy, an adverse safety profile, or manufacturing challenges, but we have some new preparations coming. Because there's such variability in the pharmacokinetic and pharmacodynamics of the different preparations, we actually have to treat them as individuals when we decide how do we dose, how do we monitor. And although it's going to be an unphysiologic growth hormone profile, the evidence so far doesn't show that that compromises the treatment efficacy or the safety. And so we have good data saying safe and effective, but we're going to need long-term registries to look at the prolonged exposure to this unphysiological growth hormone profile. So we need long-term safety data for metabolic, cardiovascular, neoplasia concerns. We have to figure out what's going to be the requirement by the regulatory agencies, by the public. We want to know what's the final height data and shouldn't it actually be better than daily, and how are we going to follow that? And I think I mentioned some of the practical implications as we went. How do we adjust the dose? In the clinical trials, we're not going to get much of that information because they're sticking to a dose unless there's a elevated IGF-1 or a low IGF-1 or an adverse event. Will anti-growth hormone antibodies impair growth? None of the studies so far have suggested that that's the case, but I think we need more data. When should we actually measure the IGF-1 for both safety and efficacy? And then what's probably more important in the US is how will it be covered by the insurance program? Are they going to be willing to say, we'll pay for this because adherence and effectiveness is going to be better? Or do we need to show that it improves the quality of life of the children to get less shots? And will there actually be an ability to prescribe long-acting growth hormone for growth hormone deficiency after the clinical trial? Will that actually give us the ability to prescribe it for other indications, both in the US and elsewhere? Because some of the molecules are so similar to daily growth hormone that they may be able to have the same label. So I'm sorry that was a fast review because I was trying to fight the technical difficulties at the beginning, but I'm going to be happy to answer any questions and have further discussion with the audience and with Dr. Rosenfeld. I always like to say that even if we don't grow, we are still perfect. And acknowledge Kevin Yuen and Eric Velasquez, who were my partners in publishing a recent review of long-acting growth hormone preparations in the Journal of Clinical Endocrine and Metabolism, and my email for people who don't get their questions answered today, who want to ask questions afterwards. So thank you very much and look forward to your comments and questions.
Brad, Ron here. Thank you for a superb lecture, very, very comprehensive. And we actually have many questions and comments coming in. But let me kick it off, if I may, with a couple of questions for you. So the first one is you've discussed the challenges of using IGF-1 measurements with long-acting growth hormone. Some people measure the peak, some people measure the troughs, some people make efforts to come up with some kind of calculation of the mean or the integrated IGF-1 level. What do you personally believe is the most meaningful based upon what you've seen so far?
I think that we need to know what the troughs and the peaks are, to understand the pharmacokinetics and the exposure. So I think I'm happy that they're gathering that data, but I really think from a practical perspective, the calculator where we can determine an average value will be so important for dosing and efficacy. I don't think we've seen any data that tells us for certain that a high IGF-1 for 1 or 2 days during the peak has a deleterious effect, a safety adverse event effect. And if we're reducing the dose because of a high peak, then we may sacrifice efficacy. So I really do think that a well-informed calculator will give us that best information to judge from, because unfortunately the kids don't come to our clinic on any specific day compared to when they get their injections, and so that'll be key.
So Brett, I agree with you. I think the most meaningful and perhaps the most challenging would be to come up with some measure of the average or the integrated IGF-1 over the course of the week. But let me throw out a comment and ask you to respond to it. In reviewing the data that are emerging from all of the clinical trials, it appears to me that to obtain a satisfactory growth response with a weekly long-acting growth hormone, it seems inevitable that many patients will, for several days of the week, have IGF-1 levels that are above some arbitrary definition, totally arbitrary, of what we consider safe, whether that's plus 2 SD or plus 2.5. Do you agree with that?
I do agree that when we look at the peak values and the growth response, that having enough of the long-acting growth hormone to get a good growth response, we have to have IGF-1s above +2 or +3 in the peak values for a couple of days. I think that is inevitable based upon the release profiles of the molecules that we currently have available. None of them seem to have a very flat pharmacokinetic or pharmacodynamic curve. So if there's a slower release process, it hasn't been yet studied.
So along those lines, do you believe that it's feasible that we will see a longer acting preparation, one that can be given monthly or annually? Is that feasible, do you believe?
I think it is feasible. I don't know, this is the second foray into or third, if you will, foray into the development of long-acting growth hormone molecules. I think people would like a longer value. Less shots would be better as long as they're as safe and effective. But I think the technology exists for that. Whether it gives big excursions or a slow pattern is the difference as to whether I think people will want to use it. Because if a once a month just has a higher peak, I don't think that would be an advantage. I don't think it'd be a good profile.
So several people have asked you to comment on immunogenicity. Is this an issue with long-acting growth hormones? And if so, what assays are relevant?
So I think the assay question is going to be important because the FDA is requiring drug-specific assays to be developed to say how can we know for certain that this new molecule is not having antibodies that are either drug specific or interfering. And I think the interference is the big question. The neutralization is the key. And so far from the different companies, we haven't seen a significant increase in neutralizing antibodies. There have been some molecules that have increased the number of growth hormone antibodies in general. And I haven't seen any that have suggested that it impairs treatment response, but I think it's still early days in terms of what does that data look like and what does it mean in the long term, and will be important for us to have access to drug-specific antibody assays to answer that question, because if we have children who aren't growing well on the treatment, that may be one of the things that we need to assess.
So I agree, Brad. Let me add to that a number of the long-acting preparations have actually dropped out of the race because of the appearance of neutralizing antibodies, although, as you said, those antibodies have not necessarily been associated with growth attenuation, but just the specter of a neutralizing antibody has been of concern. So my general comment, and please tell me if you agree, is that we should not necessarily be concerned about anti-growth hormone antibodies, which apparently show up with every preparation, but neutralizing antibodies still represent a potential issue.
Yeah, I agree with that. And I think that the anti-growth hormone antibodies, in our clinical practice, unless you have a GH gene mutation, we haven't seen growth hormone antibodies be significantly impairing treatment responses. Even going back, if we had to put someone back on daily growth hormone, there's the potential that it could impair activity. But they haven't been clinically relevant in our practice. And so I think with the new molecules, we have to confirm that that's the case as well.
Several people have asked you to put on your prediction hat and tell us what you think the cost will be for a long-acting growth hormone.
It'll be expensive. My prediction is that it'll be slightly more expensive than daily growth hormone. I don't know if that's 10% or 15% or what that will be. But ultimately, it's going to be the payers who decide whether or not they'll accept that cost for the benefits that we're seeing with the long-acting growth hormone. And my prediction hat is that they will get approved and they will get formulary status on different insurance providers or government-based systems, but it's going to be not if it won't happen if it's a significant premium. That cost will be part of that decision for the payers. That could actually impede progress in terms of transition to long-acting growth hormone. And so that's my main concern in the US is, will we actually be able to prescribe it for our patients when it's available?
But Rick Levy has asked, how important do you think growth hormone pulsatility is, and are we going to lose something by using a long-acting growth hormone?
So I know that a number of experts in the field have been concerned about the lack of pulsatility, the lack of change in growth hormone when we're eating or not eating may have significant metabolic effects. We know that daily growth hormone wasn't physiologic to begin with and has had both positive linear growth and metabolic effects in children and adults. So I think the long-acting once-weekly products will be less pulsatile, but I don't think that they'll be less efficacious in the long term. But if we had a way to mimic what the body does with growth hormone, I think that would be ideal. But right now, I don't think that's feasible. And I think we've shown with daily that it's not as important as we thought it was.
So I agree with your very important point that daily growth hormone in and of itself was not pulsatile. But I would add to that when we give growth hormone once a week, we have to look at both the IGF effects and the growth hormone effects.
Yes.
The IGF is going to persist for 5, 6, or 7 days. The growth hormone, even with the long-acting growth hormone, is going to be gone after 1 or 2 days. And so the ratio of IGF and growth hormone actions, especially metabolic actions, is not necessarily going to be exactly the same as what we see with daily growth hormone. We may, in fact, end up seeing more IGF effect than growth hormone effect because the IGF is going to be persisting. And we should remember that the two hormones, IGF and growth hormone, have opposing metabolic actions. One is insulin-like, one causes insulin resistance. So I think your point about carefully monitoring the metabolic effects, even though that's much more difficult than simply looking at growth, is going to be important.
I want to follow up
Go
on that, Ron. Sorry. I think there are 2 points that should be made on that. One is the fact that we shouldn't be considering long-acting growth hormone in children with hypoglycemia because the benefit of the growth hormone part of the molecule may be gone sooner and result in hypoglycemia. So we have to look at that population and not consider long-acting growth hormone for them until we have more information about whether it may be safe or may need to be dosed differently in that group because of growth hormone being gone. The second point that I would make is it's possible that when we look at changes in BMI that were brought up by Paul Sanger with the long-acting growth hormone from Versardis, that could have been related to the difference in the timing of injection of once per month or twice per month, different from whether it impacted the access to the tissue. So as you pointed out, the growth hormone to IGF-1 ratio at different times in the profile may be more important metabolically with a longer interval between injections. So that might be one of the reasons why that pattern was seen.
Brad, several people have asked about the use of long-acting growth hormone in patients with histories of cancer.
Mm-hmm.
Do you see any particular concern in that regard?
It's an interesting question. I don't think that, we won't have any data from the clinical trials to answer that question because those populations were excluded specifically from the trials. But as a neuro-oncology provider, they're a population that needs replacement and has been through a lot and would much prefer to be once weekly versus daily. So I think it's an advance that they should have available to them. I don't think, based upon the IGF-1 profile being elevated for a couple of days and then down, that we're putting them at a different risk for recurrence or a new tumor based on the data that's available. But that's my opinion rather than based on evidence because we don't have it yet.
Do you have any concerns about pseudotumor cerebri, increase intracranial pressure when individuals receive a long-acting growth hormone that may be around for a while they're still symptomatic?
Yeah, I think pseudotumor is, a known side effect of too much growth hormone too fast is the way that I explain it to my patients. We have some people who are exquisitely sensitive to it and others who are not. So I think that there's the potential to see pseudotumor in the children receiving long-acting growth hormone, and it will take longer for the symptoms to clear just because of the long-acting nature of the medication. Um, but I don't think that we're going to see more of them in long-acting than we did in daily. I think it's just a nature of the patient's responsiveness. Just going to take a little longer to get better.
I agree with that, but it is an issue for sure.
Yeah.
So one of the arguments for long-acting growth hormone has been the well-documented poor compliance with daily growth hormone.
Yeah.
How do we know that compliance will be better with a weekly preparation?
We know from studies of other medications that have gone from more frequent to less frequent that the compliance improves. But aside from that, we don't have that data because in the clinical trials that are being done, if you're part of a clinical trial, your compliance is fabulous because you're being regularly reminded and filling out a diary or putting something in an electronic device saying, I took my medication and here's when I did it. And if you don't, someone's going to call and remind you. So I think we need the real world data, just like I demonstrated from 3 different studies of what does it look like after it's approved before we're going to be able to answer that question. Because it isn't a given that once a week is going to be better adhered or more persistent than daily, because patients will still get injection fatigue. Patients will still stop taking their medicine. It's better to me in that if Johnny forgets his medication on Sunday, he can take it on Monday. And we don't know yet, but he could probably take it on Sunday again and not have any adverse effects. He can get back on his Sunday routine. Whereas if Johnny forgets it on Sunday, we don't usually tell him to take 2 shots on Monday to make it up because we don't think it's going to help. So I think there's at least a little bit of potential for, I forgot it yesterday, I can still take it the next day. But that has to be shown in real life as well.
Well, just a few more questions.
Sure.
You mentioned that Jintrolong, the Chinese growth hormone, is pegylated.
Yeah.
Are there any other of the long-acting growth hormones that are currently in clinical trials or applying for approval pegylated or have PEG as part of their structure?
Yeah, so TransCon GH is pegylated, but it's a reversible pegylation. So the PEG protects the growth hormone molecule from degradation as a possibly subcutaneous, but probably more so intravascular depot, where the growth hormone is being released as the TransCon linker is being degraded. And so, yes, it's pegylated, but it's reversible and ultimately releases the native growth hormone. There were multiple pegylated growth hormone molecules in clinical development in the past, but I don't know of any others currently.
Are there any plans to introduce Jintrolong in the United States or Europe that you're aware of?
Not that I'm aware of. As I mentioned earlier, I got to visit China in January to give
You're the one who brought the virus back to the States.
It's my fault, yes. Actually, I was there right as it was hitting the news, so it was bad
But we're
timing.
going to call you Typhoid Bradley from now on.
I've been healthy ever since, so though I do cough once in a while. Um, so, I think they are interested in marketing it outside of China and already do so, but I don't know what other markets that they're looking at, and I'll be interested. I'm particularly interested in their phase 4 data, to see, as they've got 3,000 children on Jintrolong, how they're growing, and whether or not they're actually having lipoatrophy, which was a concern in previous studies, and the metabolic impact, as we talked about, for the ratio of growth hormone to IGF-1.
Brad, I think we're going to have to bring this superb presentation to a close.
Well, thank you.
You very kindly offered to address any questions or concerns that the audience might have via your email. Very kind of you. On behalf of the Human Growth Foundation, I really want to thank you for just really a superb, superb lecture, and remind everybody that our next webinar, I believe, is on August 25th. You'll receive email notification from the Human Growth Foundation. Philippe Bacaliao will be giving that lecture. So with no further ado, I'm going to bring this lecture to a close, allow you to take off your tie. And Daphne, any last words that you want to provide?
Thank you so much, Dr. Miller and Dr. Rosenfeld. This was an excellent presentation. We do encourage everyone to visit us online at hgfound.org. And of course, please support those who support us by going to their virtual exhibit hall. Again, thank you so much.
Okay. Be well, everybody. Thank you again.